Hello, class, and welcome to our lecture, Evolution of Populations, aka Microevolution, which is where we're going to study evolution at its smallest scale, the population level. Hence, microevolution, micro meaning small. And this is chapter 23 of your online textbook. So, guys, hopefully you have your note outline out. You could fill it in as I go through these slides if you haven't already filled it in. So first thing we have to do, cover some terminology, go over some terms to make sure we're going to understand our discussion today. And the first term we have to define, microevolution. That's what we're studying, which is the change in the genetic makeup of a population from generation to generation. Once again, it's kind of just studying how are the genes changing in a population? How are the gene frequencies, the allele frequencies changing in a population from one generation to the next? And once again, this is studying evolutionary change at its smallest scale. Remember, individuals don't evolve, entire populations will evolve. Now, when you study microevolution, you're pretty much also studying population genetics, the study of how populations change, once again, genetically over time. How are their genes changing? How are alleles frequencies changing? Now, when it comes to microevolution, we're starting to kind of think of evolution on a grander scale. So we should define what we call the modern synthesis evolutionary theory. It's a more comprehensive theory of evolution that integrates ideas from several fields, not just biology. For example, it combines statistics, biology, which is what we're kind of doing right now, but also genetics, geology, paleontology, botany, how the world is changing, how life is changing, all sort of life changing on this planet. So once again, it's a broader theme of evolution, a more comprehensive theory of evolution. Now, since we're going to be studying microevolution, evolution of populations, we should define what a population is: a localized group of individuals that are capable of interbreeding and producing fertile offspring. And guys, the key thing, fertile offspring, offspring that can themselves have babies, have offspring themselves. Now, when you study a population, you also study its gene pool. That's the amount collection of genes in a population at any one time. When you're studying a population, you look at the genes. Are there genes there for some sort of disorder? What's the gene frequency of maybe those that have blue eyes compared to brown eyes? So it would consist of all the alleles at a gene locus or location in all individuals of that population. And if only one allele exists at a particular locus, then we would say in that population, that allele is said to be fixed. For example, let's say we're looking at the entire population of AP biology students, and let's say all of them have brown eyes, then we would say that that allele is fixed. Brown eyes allele is fixed for that population. And then the last term here, allele frequency. It's just each allele, what proportion is it in that population? What's the frequency of that allele? So let's look at an example of this. Let's say we have a population of bugs that we're studying, and we have green bugs and golden bugs. And let's say we just count the alleles. So let's count the alleles for green, the green allele. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 12 alleles. So 9 divided by 12, that means 75% of this population has the green allele. Let's count the golden allele. Notice we have 1, 2, and 3. 3 out of 12 alleles. That means 25% of the bugs here are going to have the allele for golden color. And notice that's our first generation. And let's say they breed and we have our second generation, we could count the alleles again. And notice now we go down to 71% of the population has the green allele, 29% have the golden allele. Notice it changed. That's microevolution. We had a small little change. Notice we had a little decrease in the green allele, a little increase in the golden allele. That would be an example of some microevolution, a population changing slowly when it comes to its genotype, its allele frequencies. So let's get to the big point, though, of Microevolution. That is studying an equation, a theorem, some math called the Hardy-Weinberg theorem, developed by two scientists named Hardy and Weinberg. And they developed this benchmark used to measure changes in allele frequencies. In other words, they come up with an equation. They came up with math to show that evolution occurs. Now, their theorem, though, it's going to be a little bit confusing, but hang in there, states that the frequencies of alleles and genotype in a population's gene pool remain constant from generation to generation. Now, provided only that Mendelian segregation and recombination of alleles are at work. So kind of this theorem is kind of almost just saying that, hey, allele frequencies, genotype frequencies should remain the same provided that only Mendelian segregation and recombination of alleles are at work, along with some other conditions that we're going to talk about. So when we look at this, we know a little bit about Mendel. So it describes how Mendelian inheritance preserves genetic variation from one generation to the next in populations that are not evolving. So it kind of lays the groundwork for us to understand long-term evolutionary changes. And once again, I know it's a little hard to understand now, but stick with this discussion. I think it's going to make a little bit more sense as we go through these slides. Now, here's an example straight out of your textbook, how we could use their equation to figure out, their theorem to figure out allele frequencies, genotype frequencies. So let's say we have a population of wildflowers and we have 500 of them. 320 are homozygous dominant, 160 are heterozygous, 20 are homozygous recessive, and that's our white flower here. So first, you could very easily figure out genotype frequencies. 320 divided by 500, that tells us 64% of the population are homozygous dominant. We could do the homozygous recessive, hey, 20 divided by 500, that tells us that about 4% of the population is homozygous recessive, in other words, white. And then 160 divided by 500, that tells us 32% of the population are heterozygous. And then we could even figure out allele frequencies as well, where the dominant frequency allele is 80%. 80% of the population have the dominant alleles, and only 20% of the population has the recessive alleles. So notice here's one generation. We have these numbers for one generation. We know about Mendel. We could do a Punnett square to kind of predict what we should see in the other population. And notice when we do our prediction, we get the same numbers. Notice they're exact. This means this population isn't evolving. Notice all the frequencies stay the same when it comes to genotype and allele frequencies as well. So if this happens, we say that that population is at Hardy-Weinberg equilibrium. So suppose that these individuals in a population not only donate their gametes to the next generation at random, but they also mate at random. They're going to have the same allele frequency, not just the same allele frequency in the next generation, but also their genotype frequencies can be predicted from these allele frequencies. And we could now state that those populations are in a state of Hardy-Weinberg equilibrium. Now, here's the genius part of this, though. When you apply this to populations in the natural world, most of them are not at equilibrium. And if a population is not at Hardy-Weinberg equilibrium, what does that tell you about that population? That's right. They're evolving. They're micro-evolving. Guys, there's not very many populations that are Hardy-Weinberg equilibrium. One that is are usually wildflowers. Wildflower populations tend to be in Hardy-Weinberg equilibrium. Now, what's also the genius of this, we could use this equation they developed to help us figure out allele frequencies, genotype frequencies in a population. And let's apply that on this next slide. So we're going to use their Hardy-Weinberg equilibrium equation to kind of figure out some information. So first, let's set up the equation. Let's go and kind of define what each part of this equation is. So this is their equation they developed. P squared plus 2pq plus q squared equals 1. So this is always for only a trait, a gene that has 2 alleles and 3 possible genotypes. So what do these numbers mean up here? So first thing, p squared. P squared refers to those that are homozygous dominant in a population. So p squared are all those that are homozygous dominant. It's the frequency of homozygous dominant. Let's say we're using the letter A to represent our gene. Big A, dominant, small a, recessive. So p squared would be big A, big A. That's our homozygous dominant genotype. 2PQ, that refers to everyone in that population that is heterozygous, big A, small a. That's our 2PQ. And then Q squared, that's our homozygous recessive. That's everyone in the population that is homozygous recessive. So Q squared, once again, will be the frequency of small a, small a. So just to recap, P squared, homozygous dominant, 2PQ is heterozygous, and then Q squared is those that are homozygous recessive. And they have to equal 1. Their alleles, since we're only looking at 2 alleles, should equal 100% or 1. Now, the sum of the frequencies from all alleles must also equal 100%. So we have another equation, P plus Q equals 1, where P is just the dominant allele and Q is just a recessive allele. And they must also equal 100% or 1. So just to make sure we're not confused, these up here are genotype frequencies. Big A, small a, small a, small a. Once again, homozygous dominant, heterozygous, homozygous recessive. This right here, this is allele frequencies. So P would just be big A, that's it. Q would just be small a. These are allele frequencies. Now, if a population, once again, is in Hardy-Weinberg equilibrium and its members continue to mate randomly, generation after generation, allele frequencies, genotype frequencies remain the same. But once again, when you apply that to most populations, that's not going to happen. They're evolving. Now, like I mentioned, we could use this equation to do some population genetics and actually figure out frequencies of certain possible disorders that we see in a population. So let's apply that on this next problem. So let's say we have some information, and let's say we're studying this disease called phenylketonuria, aka we usually just call it PKU for short. It's a disorder where we don't break down an amino acid called phenylalanine, and it could cause damage in the brain. And let's say we're given some basic information. We're given that 1 in every 10,000 babies in the US is born with PKU. So guys, with that information, we're already given something. Now, let's say we're also told that PKU is recessive. So PKU is recessive. So babies with this disorder are homozygous recessive. That's going to be Q squared. Q squared are all those that are homozygous recessive. That means that they have PKU. And if we know that 1 out of every 10,000 babies in the US is born with PKU, we actually know what Q squared is. Q squared is going to be 0.0001. Now, how did I get that number? I divided 1 by 10,000. 1 by 10,000 babies is PKU. That's our homozygous recessive. That's Q squared. Q squared equals 0.0001. Now, if we have Q squared, we could figure out Q, just the recessive allele frequency, pretty easy. If you square root it, if you square root this, both sides, we get Q equals 0.01. And that is the allele frequency, the recessive allele frequency. So this is the homozygous recessive, and this is just the recessive allele frequency. Now, if we have Q, we could figure out P pretty easily, the dominant allele frequency, because remember, P plus Q equals 1. So we could solve for P. So P then would equal 1 minus Q. And hey, we already have Q up here. So P minus 1 minus 0.01 equals 0.99. And 0.99, that is our dominant allele frequency. So that's our dominant allele frequency. This is our recessive allele frequency. And this once again was our homozygous recessive genotype over here. Now, let's say we wanted to find out, okay, what part of the population are carriers of PKU? They don't have it, they just carry it. In other words, they're heterozygotes. Well, we know that in our equation, P squared plus 2PQ plus Q squared equals 1. Remember, those that are 2PQ, those are heterozygous. So we have P and Q. So 2 times P, which is 0.99, times Q, which is 0.01, multiply all those, you get 0.0198, round to 2 decimal places. And that would tell us that about 2% of the US population are carriers for PKU. Once again, 0.02, we want to know a percentage times 100, that's 2% of the population are PKU. So really cool that we could use this to figure out kind of these frequencies in a population. And let's say This was the first generation. And let's say we studied this in the next generation, let's say in about 5 or 10 years. And let's say it increases from, oops, sorry about that, from 2% all the way to about, let's say it jumps up to 8% in a population. Boom, microevolution is occurring. That population is changing slowly over time when it comes to allele frequencies, genotype frequencies. Now, for a population to be in Hardy-Weinberg equilibrium, which means they're not evolving, there has to be some conditions that have to be met. First is large populations. It has to be a very large population for it to be in Hardy-Weinberg equilibrium. It also has to be isolated from other populations. There can't be any migration. Individuals can't be coming out. There can't be a leaving. They have to be isolated. Sort of like how you're isolated right now in your home. You're not allowed to really get out there because of our social distancing that we should be practicing. Also, no mutations can occur. No mutations can occur in that population. All mating has to be done at random, which if you think about that, that's kind of weird. And then last one, no natural selection can occur. All genotypes have to be equal in their survival and reproductive success. Differential reproductive success can't alter genotype frequencies, so there can't be any natural selection. So when you apply all these 5 rules to real populations out there, are any of them really going to meet all these 5? No. And if they're not meeting these, guys, they upset Hardy-Weinberg equilibrium, which means they're evolving. They're micro-evolving, boom, using math to show that evolution occurs. And actually, from these 5, when it comes to animals, at least just animals, which one do you think they break the most? If you guessed random mating, you guessed right, guys. Most animals do not mate at random. There's a lot of what we call sexual selection. I mean, are you going to mate at random? Oh, I sure hope you're not mating at random. That would just be weird. I think, females usually in the animal population are very choosy about who they end up mating with. So something we'll talk a little bit more about in our next set of notes. So, guys, that pretty much finishes our discussion over microevolution, at least the first part. And guys, we got some terminology down. We know microevolution, what that means, population genetics, modern synthesis, a broader view of evolution. But guys, the big thing here, Hardy-Weinberg, that's the key thing. We know the theorem, we know the equation. You don't have to memorize the equation, guys. It'll be given to you on exams, on quizzes. It's just a matter of being able to use it. And guys, it's going to take practice. This is not the easiest thing. It's a little bit complex. So we have a practice problem, and I'm going to work this out. Now, stay tuned. Don't leave. Guys, I'm going to make another video, and I'm going to attach it right after this so you could see how to work out this practice problem where at a locus with a dominant and recessive allele in Hardy-Weinberg equilibrium, 16% of the individuals are homozygous for the recessive allele. And let's say we want to find out what is the frequency of the dominant allele in the population. So think about that. Stay tuned. I'm going to once again record an additional video, record it and add it to this so you can see how to work out that problem. So guys, thanks for listening and have a great day. And once again, stay tuned. Okay, class. So once again, I am going to work out the practice problem at the end of our microevolution lecture. So notice the practice problem says at a locus with a dominant and a recessive allele in Hardy-Weinberg equilibrium, 16% of the individuals are homozygous for the recessive allele. They want to know what is the frequency of the dominant allele in the population. So first thing, I want to set up my equation. So I have P squared plus 2PQ plus Q squared equals 1. And remember, I want to define what these are. So once again, P squared, that's my homozygous dominant. So that's kind of like my big A, big A. My 2PQ, it's all those that are heterozygous. My big A, small a. And then Q squared are all those that are homozygous recessive. Small a, small a. So hopefully you guys see that pretty well in this video. So those are my genotype frequencies. Remember my other equation though, I have P plus Q equals 1. These are just allele frequencies. P is the dominant allele, big A, so that's just my dominant allele frequency. And my Q is just my recessive allele, so that's my recessive allele. So once again, these are genotypes and these are allele frequencies. Big difference there. So notice what the problem tells us. It tells us that 16% of the individuals are homozygous recessive for these alleles. So they're homozygous recessive for the allele. So we got to figure out, what are they giving us? They're giving us something. And notice, if 16% are homozygous recessive, hopefully you have guessed or know that that is referring to Q squared, not Q. Q is just a recessive allele, but notice it's homozygous recessive. So that tells us that Q squared equals 16%. We can't deal with percentages, so we're going to break that down to 0.16. So Q squared equals 0.16. Now, that's what I have. Now, what do I want to figure out? Notice what the question is asking. It wants to know the frequency of the dominant allele in the population. So what are we trying to find out? If we want just the dominant allele in the population, we're looking for P. So P, that's what we want to find out. So P is what we're trying to get. Now we have Q squared is 0.16. Well, guys, from here, Q squared equals 0.16. We should be able to figure out Q pretty easily by doing what? Well, notice you could take the square root. Take the square root of both sides and you get Q. And if you have your calculator handy and you dare do the square root of 0.16. Oops, let me get that right. Notice that is going to be 0.4. 0.4. Q is 0.4, and that is our recessive allele. So now, is that what we want? No, we want P. But guys, notice our equation. P plus Q equals 1. Let's plug that in. So we have P plus 0.4 equals 1. Well, now we just solve for P. P is going to equal 1 minus 0.4. And then if you do that, P equals 0.6. And is that what we're looking for? Yes, that's our answer that we're looking for. So we could say that 60% of the population have the dominant allele. And that's what we were looking for. So once again, 60% of the population have the dominant allele. In other words, just big A in this case. So that's our answer. So hopefully this helped out. Now guys, now it's your turn. Work some microevolutionary problems on that problem set and see how well you guys do. Okay, so hopefully this was helpful. Guys, hopefully I will see you soon on a Zoom meeting, or once again, post your questions on Edmodo or on the next Zoom meeting. Have an awesome day.